Hello, everybody. My name is Dave Harwood, and for our third journal club, I'll be presenting a paper that I found that was recently published, hot off the press, entitled The Integration of 168,000 Samples Reveals Global Patterns of the Human Gut Microbiome. So the bottom line up front is that researchers created the largest compendia of microbiome data to study global variations within the human gut microbiome. And in this process, they revealed regional differences of the most common genre of microbes and investigated opportunities for future research in order to address underrepresented world regions. Now, why should we care about this field? Well, overall, the research enhances our understanding of the gut microbiome's impact on health, and it provides a path forward for innovations in medicine and public health. Particularly as we look at these bullet points, we see that for health insights, our gut microbiome plays a crucial role in our overall health, influencing digestion, our immunity, and even mental health status. As many of our classmates have pointed out in previous discussions, the gut-brain axis plays a vital role in determining the composition and diversity of gut microbiota with mental health. So understanding global patterns can help us identify factors that contribute to health and disease on a very large scale. Now, this also has a place for personalized or individualized medicine where we can recognize regional differences in these microbiomes and develop more effective personalized treatments and even some dietary recommendations that are fit for specific populations. And then for disease prevention, and I think this one's kind of interesting because a lot of papers presented this semester have linked changes in composition and diversity of gut microbiota with disease. So one of the biggest hopes of this paper is that insights from this research can lead to better prevention and management of diseases such as obesity, diabetes, inflammatory bowel disease, which are linked to gut microbiome imbalances. Again, really just connecting dots between chronic inflammation, disease, and that composition and diversity of the microbiota. There's also been significant scientific advancements, and hopefully this paper provides a valuable resource for further research that'll be able to give scientists the ability to explore new microbial taxa and their functions, and hopefully lead the way for new groundbreaking discoveries. And then as we look at things on a global scale, we do want to draw particular attention to undersampled regions and emphasize the need for more inclusive research so that we ensure that health benefits from microbiome studies are accessible to diverse populations everywhere around the world. And so essentially, researchers wanted to know what the global patterns and variations are in the human gut microbiome. And through their efforts, they built the largest and first compendia of data. And I believe that the researchers feel like these goals were achieved for those aforementioned reasons. So getting into a little bit of the background that led to the formation of this large compendia of data. So first, we have to kind of go back to the beginning and talk about advances in the field of microbiome research and bioinformatics. So one of the things that we learned in one of the first modules this semester, and in particular in I think module 5, was we discussed the Human Microbiome Project, which was a huge initiative by the National Institutes of Health to lay the groundwork and foundation for mapping out the diversity and functions of gut microbiota. The projects involved with the Human Microbiome Project and other metagenomics projects led to the revelation of intricate relationships between gut microorganisms and their human hosts. And so this kind of highlights the role in our immune systems, the individualized medicine approach, metabolic regulation, and overall health. There's recent studies that have focused on an understanding of the variability of the gut microbiome across different populations. And I think we even, as a class, discussed one in our intro to Journal Club. Advances in metagenomic sequencing technology have enabled us to have more detailed profiling of the microbiome. And this has given us insight into new microbial taxa and function that were otherwise previously unknown. So we're now exploring the gut microbiome's impact on conditions such as obesity, diabetes, mental health, and really trying to develop personalized, individualized medicine approaches based on microbiome composition. Now, despite some of these advances, there are challenges that still present themselves, including arriving to the definition of what is a healthy microbiome. And in order to do that, we have to understand strain level differences and translate those findings from the bench in the lab to clinical practice at the bedside. The integration of large data sets like the one in this study is going to be super crucial for addressing these challenges and also advancing our understanding of the gut microbiome's role in health. So the broader implications of this research are that it's still a relatively new area of research that until recently lacked a large central compendia of data for researchers to utilize as a central resource. So the overall goal here is to standardize the metadata analysis from samples collected from a variety of projects for a variety of reasons. All of those projects had very specific purposes and hypotheses. And through that, we created this large compendia of data. So before we get into research findings, we kind of have to talk about, well, how did they build this huge data set, this huge compendia of data? So this is a gross oversimplification of these steps, but essentially the researchers started with retrieving a little more than 245,000 samples from a little more than 1,400 bioprojects from NCBI, and this was just publicly available information to the researchers to begin the compilation of their database. They then excluded projects that had less than 50 samples, and then they further excluded projects that utilized pyrosequencing data that was dissimilar to Illumina, and there's a reason for that I'll come back to. Further exclusions were made for projects containing errors through the process of manual curation, and then they excluded samples that failed data 2 QC parameters, and that's a tool that we'll talk about in a little bit more detail in a little bit. They also excluded projects that had more than 2 sequencing platforms. They focused mainly on Illumina. So if we think back to one of our first modules this semester, and we think about the technology that's been developed in the field of microbiome research, Illumina was one of the first widely accessible technologies, and over time has become one of the most cost-effective for researchers around the globe to use to sequence and characterize microbial taxa within the human gut microbiome. So in order to avoid batch effects statistically as they compiled their data, they really wanted to make sure that the source data was fairly consistent across the board, and so they stuck with a process of sample processing that could be reproducible irrespective of whatever global region the data was coming from. So that was really the main push for using Illumina. And then they finally arrived at a final number of samples that was a little over 150,000, and that was after they filtered some low read counts. So this is figure 1A from the paper, and it's essentially what I just talked about on the previous slide, but it's a nice illustration that shows what they refer to as a single pipeline to arrive at this final compendia. And what we see here is that, they start with a little over 245 1,000 samples, and they get down to about a little over 168,000 for their final compendium that's published online for everyone to utilize for future microbiome research. Now, the presented analysis and their research findings that were in this paper after they had created this huge database actually comes out to a little over 150,000 samples after they took out some low read counts and some unidentified taxa. So just a quick word on that data 2 QC exclusion. So the data 2 QC parameters, it's essentially a QC or statistical tool that helps to improve accuracy and resolution. What it does is it identifies and counts the different types of microorganisms present in a sample by analyzing their DNA sequences, and then by using statistical methods to correct for errors produced during DNA sequencing, it's able to order and group similar sequences together in order to identify more specific types of microbes. And what this does is it helps provide higher resolution of the data. So we talked about how they put the largest data compendium together of sample information sequences, microbial taxa of all microbiome research that is publicly available. What did they find? So in brief, this project helped to create one of the largest unified data sets ever compiled, the largest unified microbiome data set ever created, consisting of over 168,000 samples through a single pipeline that we just saw in Figure 1A. This research revealed significant variations globally within the human gut microbiome, with certain genera of microbes like Firmicutes being almost universally present across all global regions. What's really interesting is they use a different name for the Firmicutes phylum, and we'll get into that into more detail later. Now, they did find differences when they started comparing these global regions, such as the relative abundance of about the 65 most common genre of microbes. And those varied significantly between these different world regions. The study also highlighted that regions like Central and Southern Asia have unique microbiomes that are vastly underrepresented in current research, and that these regions likely harbor hundreds of taxa that have yet to be discovered. And this is mostly attributed to undersampling within research. So this kind of brings us to the last point, that the researchers emphasize a growing importance for the need to include diverse populations within microbiome studies so that we can have a comprehensive understanding of equitable health benefits on a global scale. So I think the overarching goal here and hope is that this compendia, along with the particular research team's findings, allow for future studies on the human gut microbiome to explore areas such as individualized medicine and disease prevention. Okay, so here's the first figure from the paper, and what we see in the upper left-hand corner is Figure A, and we've already talked about that one in detail, but essentially it's showing us that single pipeline that they used to take samples from that initial over 245,000 from a little over 1,400 bioprojects and getting it down to about 168,000 samples. Now in block B, we see a histogram that's showing us the distribution of reads that were classified in each sample. And so that x-axis shows us the number of reads in each sample, while the y-axis is indicating the number of samples with that number of reads. And this is just a normal distribution. It's showing data that is respective of the total compendium. So not just global region, but the entire data set altogether. And that vertical gray line, right, let me put my laser pointer on. So right here, that indicates the median read count. Now, C through E are probably some of my favorite data represented in the entire paper. It's not the most important, but it was kind of my favorite. And basically what we see is the rank of the various genre broken down by various level of taxonomic classification. So in Block C, we see a breakdown of various phylum and how they rank in terms of composition. And then with Block D, it's the class of organisms. And then in Block E, we see the order. Block F is a density plot that's basically showing the relative abundance on the x-axis versus the samples that have been read. And so there's a gray line that indicates all other phylum, but these lines on Block F came from the 5 most prevalent phyla in the data set. And they use the same colors here in F as what was identified in C through E. And I'll go ahead and preface for everybody, I'm partially colorblind, so if I refer to a certain color and I'm actually incorrect about it, I do apologize. I do the best I can with a lot of scientific papers, but just bear in mind that sometimes I misidentify colors because I can't see them. But color deficiencies aside, in Block G, no worries there, it's black and white. We see a histogram that shows the distribution of the Shannon diversity observed in the entire compendium. So this is essentially showing diversity of all samples, irrespective of global region. And then once again, we have this gray line, which is showing the median for those reads. And then Block H is the results of a rarefaction analysis where simulated compendiums of various sizes were generated repeatedly and then evaluated for the taxonomic richness. So what we see is unique taxa against number of samples. And so we can see a breakout based on taxonomic order and how that looks. Essentially, this Block H is showing reproducibility according to the varying size of the compendia. Okay, so here's figures 2A and 2B. And what we see here, which is not necessarily surprising, is the prevalence of the number of samples within the compendium. So what we see is Europe and North America dominate the data set, and it was something like 60.5% of all samples came from Europe and North America, followed by Eastern and Southeast Asia, Sub-Saharan Africa, etc., with Latin America and the Caribbean being the least represented global region in this data set. And then if we look at Block A, what we see is global regions, and these are the same color codes that were in that are listed right here in Block B. And then the bottom part here is a distorted world map that just kind of shows the overall dominance of the global regions and essentially how they compare against each other. So here we have a very busy slide, but this is probably one of the most important figures in the entire paper, in my opinion. And this is basically showing those differences in microbiome composition between each global region. And so we're going to break down each portion of this figure in a little bit more detail. Okay, so a closer look at figure 4. We'll start with figures 4A and 4B. And what we see here in figure 4A is the number of unique taxa discovered in subsamples of various size from each world region. And so what we're looking at is each point on these curves represents the average number of unique taxa that were identified in a subsample from a given region. And this was over 1,000 repetitions. So your x-axis is indicating that number of microbiome samples selected, and your y-axis is going to be the number of unique taxa identified within those samples. And then the color for each curve corresponds to the region that's identified with the same color. Now the inset that's within this graph uses the same x-axis and color scheme, but it's showing the average number of taxa discovered per million reads on the y-axis. So it's just slightly different, but it's the same information. And then in figure B, we see a bar chart that's showing the number of unidentified taxa in each world region at every taxonomic level. So we've got, genus, family, order, class, and phylum, and then each one of these bars is a different world region. And the colors for these world regions correspond to the colors that are labeled here. What I think is really interesting is that we can see Latin America and the Caribbean doesn't have, in terms of numbers, a whole lot of unique taxa being identified based on the fact that there's not a huge sample, right? And remember, this was an underrepresented global region, but what we see in figure 4B, excuse me, in figure 4B, is Latin America and the Caribbean have the highest number of average unidentified taxa per taxonomic group. So kind of interesting that it would dominate in each taxonomic group, but because of it having the least samples, it tends to be the lowest in comparison to other world regions. Okay, and then figure 4C is going to show us each individual global region that makes up part of the overall data compendium that's been assembled. Each line associates to a different microbial phylum, and we can see that there are differences in the composition of these phylum across global regions when they're compared against the number of samples. What's really interesting here is this Baculata, I'm probably not pronouncing that right, was uniformly consistent across all global regions in terms of relative abundance. We talked about Firmicutes taking on a different name in this paper, and so the Baculata phylum was a new name. I'm not saying that this paper coined the term, but it utilized this term in place of Firmicutes. And then likewise, this Bacteroidata phylum, you may find Bacteroides as a more familiar term. So just, same faces with a different name. And then here in figure 4D, this is where we see a stacked bar chart that's going to show us the relative abundance based on world regions. So it's kind of difficult to see, but this bar on the bottom has, it might be easy for other people, but there are different colors, and these colors correspond to those varying global regions that were previously displayed in figure 4. And so each column within this stacked bar chart is an individual sample, and the colored segments indicate the relative abundance of a given phylum in that sample. And so they were ordered first by world region, indicated by that colored bar here on the x-axis, and then by relative abundance for the 5 most prevalent phyla. And so I can't see it, but the Baculata is the light blue segments in each part of this data set. It's dominant for each region But what was interesting that the paper noted was that regions from Central and Southern Asia have a relative abundance of actinomycetata that was labeled with green segments that was more prevalent. And these regions from left to right, so this first region here is Australia and New Zealand, and that's followed by Central and South Asia, and then we have Eastern and Southeast Asia, and then we have this huge section which is all the samples that came from Europe and North America, and then this tiny, tiny sliver is Latin America and the Caribbean, followed by North Africa and West Asia, and then Sub-Saharan Africa. So we can see here that, Latin America and the Caribbean, super underrepresented population within this entire data set. So to kind of summarize and put a pretty bow on this, we'll go into a little bit of detail of the summary of the findings. So coming full circle, the researchers put together the largest data set of microbiome data that had ever been assembled. This data set illustrated differences between global regions, and those differences were identified by changes in microbial composition. It also shined a light on the undersampling of certain regions, which may misrepresent findings. So here are some caveats that I think everybody should take away from the findings, and these were caveats identified in the discussion of the paper. So you don't want to draw conclusions from a broad range of samples that were collected for a variety of reasons and research aims. This goes back to the fact that these samples don't have associated ancillary information with them, such as health status, dietary regulation, disease status, et cetera, et cetera. And so that kind of leads into the second bullet point here, and that's that the data in the compendium doesn't have this information. So you kind of have to, when making such large, analysis of such large data sets, you have to kind of take into account what was the purpose for which the data was being collected in the first place. And then you have to, just bear in mind that you don't have this ancillary information coupled with it. So you really don't want to draw, you don't want to paint with a broad brush and draw these wide-ranging conclusions from, a wide breadth of samples collected for a variety of reasons. And then I think most importantly is that this data compendium is not addressing disparities with respect to that underrepresentation of samples within global microbiome research. So there's still a ton of work that has to be done in order to provide funding, infrastructure, technology, and really get research moving in certain areas of the world where the data is lacking. So 5 questions to kind of help everybody get started with the discussion. How do regional differences in the gut microbiomes affect or impact diseases in different populations? What are the potential health equity implications of underrepresented regions in microbiome research on a global scale? How can findings from this research be applied to individualized medicine in specific populations? And then what new taxa were discovered in this study, and what are their potential roles in human health? What can future research address the gaps in undersampled regions? And here are my references. And I hope everybody enjoyed my presentation. Thank you very much for watching. Take care.